it's a repressible operon. So that means that majority of the time, the operon itself is on, but it can be repressed. So normally when there's low tryptophan, when there's not a lot of tryptophan within the cell, RNA polymerase can attach to the promoter and it can transcribe the genes to produce these various enzymes that come together to form tryptophan itself. So when tryptophan accumulates within the cell, tryptophan will actually attach to what was the inactive repressor, and it makes it active. This now active repressor then attaches to the operator and prevents RNA polymerase from transcribing the genes. An inducible operon. This means that majority of the time when there is not a lot of lactose in the cell, what happens is the repressor is on whenever there's a lack of lactose, meaning that RNA polymerase cannot transcribe the genes to create lactose. But when lactose is present, an isomer of lactose called allolactose forms, and this is the inducer. So the inducer will attach to what was the active repressor, and it makes it inactive. So when it's inactive, now the genes can be transcribed by RNA polymerase to form the mRNA that codes for lactose. This is a positive feedback mechanism that uses CRP. So what happens is when lactose is present in the cell, you have a low amount of glucose in the cell, you're going to have a high amount of cyclic AMP. So when you have a high amount of cyclic AMP, it attaches to the CRP and it makes it active. When CRP becomes active, it'll attach to the promoter behind RNA polymerase. Its attachment behind the promoter actually increases RNA polymerase's affinity to the operator. So this allows the RNA polymerase to transcribe these genes and create more mRNA than it would without the CRP. So when you have a high amount of glucose in the cell, you're going to have a low amount of cyclic AMP, meaning that there's no cyclic AMP to induce the CAP, meaning it's still going to transcribe the genes and it's still going to create mRNA, but not as much as it usually would with the CAP. This is RNA splicing. So this entire thing is called a spliceosome, and inside of the spliceosome you have the small RNAs. So this is the mRNA, and these are the exons, and this is the intron. So the small RNAs will base pair along certain parts of the intron, and then they catalyze the cutting out of the intron at the exon-intron junction. So it cuts out the intron and then discards it, and it becomes degraded. And then the exons come together and are fused together, and a 5' cap and a poly A tail are added to complete the mRNA. Okay. First, there's an extracellular signal that comes to the cell, and chromatin modification takes place, and the DNA is unpacked. Once the DNA is unpacked, it's ready for a transcription. So you'll have your primary transcript, and that includes the introns and the exons on the transcript. But then this goes through RNA processing, where the RNA is spliced. So now you're only left with the exons. So it adds a 5' cap and a 3' poly A tail, which actually helps it to stop being degraded when it enters into the cytoplasm from the nucleus. So then it's transported to the cytoplasm, and the mRNA are transcribed into polypeptides. And after this, the mRNA is then degraded. So this polypeptide becomes an active protein through protein processes. And the active protein then transports to its cellular destination, and then the protein is degraded.